Hi, I'm Ahmet Denli from Cold Spring Harbor Laboratory Press and Genome Research. Last week, we had the 85th Cold Spring Harbor Symposium, and the topic this year was biological timekeeping. Joining me today is Erik Herzog from Washington University, St. Louis in the United States. Thanks for being here and welcome back to the Cold Spring Harbor in cyberspace.
Thanks for having me, Ahmet.
Before we talk about research, I'd like to briefly ask you about your background, where you're from, your path to becoming a scientist and a neurobiologist working on the circadian cycles.
Sure. I grew up in Wisconsin in the middle north of the United States. I am the child of a stay-at-home mom and a physician father. We grew up in the woods, so a lot of my life was spent exploring the great outdoors, catching critters and keeping them captive for as long as I could until they were too big and had to be let go. I went to college in Wisconsin and then North Carolina, and then did my graduate work in New York and Massachusetts. And all along the way, I thought I was going to be a marine biologist. I was dedicated to being a marine biologist right up until I became fascinated with circadian clocks in a marine organism, horseshoe crabs. And I was studying their vision and discovered that they could see equally well day and night with my PhD advisor, Bob Barlow. And it was then that I switched from studying vision and marine organisms to studying biological clocks, circadian rhythms. And I still thought I'd be a marine biologist. I was working in a lab that worked on snails with Dr. Jean Block. But it became pretty clear to the lab that the snail was no longer the best model system to study it in, and we switched to studying in mice. And I made my name in studying biological clocks in the suprachiasmatic nucleus of the mouse, and got my job at Washington University in St. Louis, studying circadian rhythms. Now, about as far as you could get from an ocean is studying daily rhythms in mice. And I think I can now say I'm no longer a marine biologist, but that's my path.
It sounds very interesting. So on Thursday, you told us 3 interesting stories on circadian synchrony. And you were covering both mechanisms and potential applications. I know it was a full talk, but would you mind giving us a brief summary of what you talked for those who might have missed it?
Yeah, I think the basic premise of my talk and the session that we had with 4 excellent speakers was really on understanding how time of day is communicated between cells to generate circadian rhythms that are coordinated in rhythms and physiology and behavior. And the 3 stories I told were about how moms and fetuses synchronize their daily rhythms during development, how the body generates daily rhythms in hormones like glucocorticoids. And the 3rd story was on how when we have brain tumors, those also synchronize to the host to have daily rhythms that may become targets for drug therapy, where they're more sensitive at one time of day than another.
So yeah, that was a quite broad talk. So starting with the fetal mother interactions, you mentioned that the PER2 cycles are potentially involved in successful pregnancy. Do you have any mechanistic insights into the function of this synchronization and how it works?
I'd say at this point, no. It's mostly phenomenological, where circadian mutants have been shown to have difficulties in reproducing. For example, the clock mutant actually has inappropriate uterine contractions at the time of delivery. The mechanisms of those, how circadian rhythm disruption, whether it be genetic or environmental, and how those impair pregnancy and delivery, I think are not fully understood and pretty can happen at many levels of biology. In our case, we were interested in just showing when the rhythms in the fetus synchronize to the mom. We found out that that actually happens very early in pregnancy. We were surprised to see that that happens sort of 50% of the way through pregnancy. And we've got an association in humans showing that disruption of circadian rhythms, women who have irregular daily schedules or irregular daily rhythms in hormone secretion, are at risk for preterm birth. And the next step is to understand more about how those mechanisms that are normally there become disrupted and can contribute to poor birth outcomes, and also to design interventions to try and rescue where rhythms are disrupted. And many of us have irregular daily schedules, right? It's hard for us to keep a regular daily schedule with today's social demands. So designing interventions that can help protect against the negative consequences of chronodisruption.
So at the interface of maybe birth and later life, I mean, this might be a question a little bit out there, but is there any circadian rhythm to the proteomic or metabolic content of, say, mother's milk?
Yeah, actually there is. There's mother's milk in the morning contains hormones like cortisol. Mother's milk in the evening contains hormones like melatonin. They have different constituents. It would be really fun to see breastfeeding moms divide their freezer in half, where they store night milk and day milk, and they deliver their milk according to the baby's time of day. But there's really been very little on this. It's just been an observation that milk has different components and nothing else.
Interesting. And another question that might be a little bit out there is, so clearly the mother and the fetus are intimately physically linked. Is there a social component to circadian synchrony? In other words, you know, do the two organisms have to be intimately physically linked, or being in a social group otherwise might have an influence?
That's great. So here we're focused mostly on events that are happening after birth. And in adult humans, the evidence for social interactions synchronizing circadian clocks is very weak, that this is not a very strong cue. So for example, some blind people struggle to synchronize to local time, and they wake up on subsequent days an hour later than they did the day before, and they sort of drift through time. Some blind people seem capable of using social cues from roommates and partners to help them synchronize to local time. But it seems to be a relatively weak signal, both in humans and in mice. Social cues are not very strong. But maybe we're using the wrong social cues, and there may be ways to enhance social entrainment. In fruit flies, there's pretty good evidence now that they can synchronize their circadian clocks through social cues. And in fact, it can be a very potent cue, for example, in social organisms like honeybees, to synchronize their circadian rhythms. But in terms of mom and her newborn child, there's not much known, but we actually have a grant that we're about to submit on this very topic. We're interested in, if mom has disrupted circadian rhythms, does that translate to disrupted circadian rhythms in the children? And could dealing with mom's circadian rhythms be an intervention that would help the kids?
I mean, that sounds very interesting and very much related to the human experience.
Yeah. And also, just as sort of a sidelight that I'd like to make sure we bring up in this conversation is, it disproportionately affects certain aspects of our society. So people who are of lower socioeconomic background tend to be the ones who work 2 jobs and have a hard time getting a good night's sleep. And we think that this affects their children's early life brain development disproportionately. And so we're hopeful that intervening with circadian rhythms is an easy intervention that can help not just mom, but the offspring in the way you were describing.
That's very interesting. Yeah, maybe we can get back to that at the end of the call if we manage to have time. So going back to the second topic you talked about presence of multiple clocks in the body, and how the combined activity might actually be more precise and adaptable. I mean, I think this begs the question, what happens when it is disrupted? Are there any diseases associated with it, or any other problems that arises from the decoupling, let's say?
This is a really important question that I think we don't know, we don't think about enough as a field. So at the meeting, there were a number of talks identifying genes associated with sleep disorders, for example. And the list of genes is almost always a list of genes that are associated with either rhythm generation, regulating sleep duration, or rhythm output. But we don't think that much about genes that are associated with synchronization between clocks. And I think those same genes that we've identified as essential for cell-cell synchronization should also be on the list as candidates for polymorphisms that could put people at risk for disrupted rhythms in internal coordination or in their ability to synchronize to the environment. So I'll give one example. VIP and its receptor, VIP receptor 2, VPAC2R, is critical for synchronization of circadian rhythms in mammals. There are polymorphisms in VPAC2R that have been associated, for example, with schizophrenia and a number of sleep disorders. Those associations have not really been tested, whether those polymorphisms are causal in, for example, a mouse model.
That would definitely be interesting. Just out of curiosity, if you look at it from an evolutionary perspective, what's the lowest organism or simplest organism you see multiple clocks? Is it known?
So there's good evidence for multiple clocks in most multicellular organisms. And even in some single cellular organisms, there may be multiple circadian clocks. So early work by Till Ronenberg in dinoflagellates revealed the presence of multiple circadian clocks in these single-celled organisms. There's also evidence in these single-celled organisms that there can be cell-cell synchronization. So the population of organisms can work together as a superorganism by coordinating their circadian rhythms. For example, some cyanobacterial events seem to be coordinated as a population. So they may be communicating with each other. There was an exciting talk at the meeting by Carl Johnson talking about seasonality in cyanobacteria. And it seems like we don't know yet whether the ability to encode seasons requires multiple clocks. And if in fact it might be all encoded by multiple clocks within a single-celled organism like a cyanobacterium, or that you get this seasonality by interactions between cyanobacteria.
It's very interesting. And the last part of your talk, you talked about relevance to glioblastoma. And you shared data that shows the xenografts actually synchronize with the host. I was curious whether there's anything known about the mechanism, the signals involved, and whether decoupling again or preventing the synchronization could be beneficial from a therapeutic perspective. Could it allow you to better distinguish between healthy cells and tumor cells in that sense?
Yeah, I think we don't know how the tumor is synchronizing to the host. Those are very new data from mice. We don't have data in humans yet. In mice, what we can say is that tumors do respond to a wide variety of signals, both neural and humoral signals. I'm very excited about the possibility of neural communication to the tumor, and we're exploring that, whether there's neural signaling during the day, for example, that's gone at night, that provides the tumor with time of day information. But we've also seen in the circadian system that signals as simple as body temperature can be sufficient to synchronize circadian clocks across the body. And that seems to be a very efficient way by which the central clock in the suprachiasmatic nucleus can coordinate timing, for example, in the lung, the liver, and the cornea, so that they're all on central time. And so it may be that the tumor is synchronizing by a multiple cues that need to be integrated, and body temperature, hormones, and neural signals will all become candidates then for therapeutic targeting. And then the second part of your question is harder for me to answer. Right now, the field is at a point where we are not in agreement on whether all tumors have or don't have circadian rhythms. All of the glioblastoma samples that we've characterized from patients, so we're now at over a dozen glioblastomas that we've characterized, they all show very nice circadian rhythms. But the literature suggests that circadian rhythms are disrupted in many cancers. It may be specific to certain types of cancer, and even within a type of cancer like glioblastoma, it may depend upon the genetic composition of that tumor. But right now, what we're finding is the vast majority of tumors have rhythms, and we think that provides a therapeutic window for us to treat them at times of greatest sensitivity and at times when hopefully we can minimize the side effects.
I mean, I was struck by the small percentage of actually therapies that take time of day into consideration. That was an eye opener in a way.
Yeah, and there I was reviewing the literature, which was really beautifully reviewed by Carla Finkelstein and John Hoganesh, and I encourage people to read their papers.
So what I'd like to ask you, it's a broad and very interesting field, and you definitely seem to be involved in multiple angles. I wanted to ask if you could share maybe a couple of recent exciting findings, be it from your lab or this meeting or the field in general, whether you could share that.
Sure. I was the president of the Society for Research on Biological Rhythms for the last 2 years. And I think the field in general is a very exciting field. We celebrated the Nobel Prize for the discovery of the molecular basis for rhythms in 2017. And I think part of the reason we won the Nobel Prize, and I say we because I really do feel like it's a big family. This is a very collaborative and interactive community. We all enjoyed that Mike Young, Mike Rosbash, and Jeff Hall won the Nobel Prize for their discoveries on the molecular basis of rhythms, because we also saw that it had a lot of translation to every aspect of biology and medicine now. I think there's a lot more interest in the medical world in the possibility that rhythms are normal and that when they're disrupted, health is impaired. So for me personally, my lab, I'm surprised that this kid who's, grew up studying vision in horseshoe crabs is now running clinical trials. So I'm very excited to be working with physicians who have drunk the Kool-Aid and said, this is worth testing. We need to know if this drug that's been approved for treating glioblastoma for over 20 years now, and we have seen no alternatives that surpass its performance. We might be able to improve that drug by as much as the drug itself had originally been described to benefit the patient. So it extends the life of a glioblastoma patient. Temozolomide extends the life of a glioblastoma patient by about 6 months. So they have a 15-month prognosis from being diagnosed with the disease till when they're going to die. And yet it looks like taking the same drug in the morning extends your life by an additional 6 months. And that's without doing anything to personalize this further, to say, optimize to your chronotype or the genetics of your glioblastoma. And so I think that circadian medicine is an exciting area for the field, where the applications are going to be very diverse. And we may be seeing big changes in the way we see ourselves being treated when we go to the doctor. And then I'm just fundamentally interested in biology, and the phenomenon of synchrony for me is really exciting. And so I'm really excited when we see new evidence for how cells communicate time of day and when they communicate it. And so I'm excited, for example, in the maternal fetal communication path, to see if we can identify how moms tell babies what time of day it is, so that they can agree on what time to deliver.
They definitely are very intimately linked to the life experience. It's a fascinating field. So as you mentioned, you're currently the president of the Society for Research on Biological Rhythms, and I believe the director of the Neuroscience Pipeline Initiative.
That's right.
To increase diversity. And you are the recipient of the 2018 Award for Education in Neuroscience from SFN, the Society for Neuroscience. So in the last few minutes, I wanted to talk to you about science and society. while the pace of scientific discovery is breathtaking, the same probably cannot be said for the efficient communication of the findings. And I could not think of a better person to ask this question. What can we do as scientists to improve the communication of our findings to the society in order to ensure the integration of science into daily life, policy, and also ensure a broad, diverse next generation of scientists?
Yeah, there are lots of things we can do as scientists to better communicate our enthusiasm for what we do and the importance of what we're finding. It would be great if everybody developed their own personal lecture on the value of basic research. I think that challenge people find surprising, that you don't understand the value of basic research. Why do I do research? So many of us would answer that immediately with, you know, I do this because I love it. I love the joy of discovery. But I think it goes deeper than that. I think we all recognize that the value of basic research is that through knowledge, we can make a better society and a better world. And so right now, for me, I think the United States is particularly focused on disparities within our cultures. And so we have people who have privilege and people who do not. And as the majority becomes the minority in this country by 2050, we have an obligation to replace the white scientists who are currently the predominant members of the scientific community with people of color who have an interest in treating the problems that society will face in the future. And so I'm really committed to finding ways to get young scientists who are not traditionally found in the scientific ranks to be excited to become scientists and to be welcomed as fully accomplished, fully capable scientists for the future. So I run that neuroscience pipeline program here in St. Louis with support from NIH and from a number of foundations where, you know, I've been really impressed by how excited my colleagues have become about participating as mentors, about how learning to be a better mentor for people who come from different backgrounds. And so the second challenge for us as scientists, if we accept the challenge of explaining why basic science is so important, the second challenge is to say, you know, I'm really not a very good mentor for everybody. I need to figure out how to be a better mentor for people who walk in this door who have not traditionally been represented in the scientific path. So one fun example, maybe just for fun, is graduate students at Washington University in the neurosciences are invited to develop demonstrations based upon their graduate work. In their second year as graduate students, we have them present a demo at the Science Center for the general public. They present to about 4,500 people over the course of about 8 hours. And during that time, they learn what it is about their thesis that really gets them excited. And I've seen students change their thesis topic as a result of talking to the public about their thesis. And they realize, you know what I'm really excited about? It's this AIM 3. AIM 1 is not actually that interesting. And so science communication can have its benefits for us as scientists.
Absolutely. I mean, I think for the continued funding and also continued influences in daily life, or better representation, increasing life quality. Yeah, absolutely. It is an important topic. And thanks for doing what you do, both in the lab and outside the lab. And Erik, it's been a pleasure talking to you. Thank you very much for being here and hope to see you in person maybe in the next meeting.
I look forward to it, Ahmet. Thank you.
Thank you very much. Bye-bye.
Bye-bye.